Antibody structure and function discoveries. In 50s, enzyme reducing agent digest IgS to smaller component. In 60, B-cell lymphoid cancer and single homogeneous type of IgS. In 69, determination of first complete AA sequence of IgS. In 1975, monoclonal technology. After that, isolation of IgS gene. Ig plus IAG cascade of molecular signaling event, B-cell activation, clonal proliferation, generation of plasma cell that produced Ig function as antibodies. The hallmark of IgS as antigen binding protein are specificity for particular epitopes, diversity as a group, position of secondary biological activities as opsonins, activating complement cascade, cross-placental barrier. Each person can produce at least 10 raised to power 8 different antibody molecules, each with distinct properties. This is the structure of the antibody, antigen binding sites and light chain, disulfide bridge heavy chain, hypervariable, here the hypervariable CDR regions, variable regions, constant regions. The basic structure: two different polypeptides, large heavy chain, smaller light chain L, large heavy chain H. Each Ig consists of 2H, 2L chain. The chains are held together by non-covalent forces. Covalent interchain disulfide bridge. HLR composed of globular domain, each about 110 AA long, single interchain disulfide bond. The AA sequence falls into 3D configuration: Ig parallel, L chain 2 domain, H chain 4 to 5 domain. The sequence of chains vary widely, especially in N-terminal domain. Other domains are constant terminal CH and Cl. Here the crystallography, antigen binding with Fab fragment. This L chain, the heavy chain, disulfide bond, the carbohydrate, the CH3 hang region, TH1 CL VL VH. Cl is only one, but CHS are 3 or more. CH1, CH2, CH3 beginning from the closest to VH. VH plus VL antigen binding site Fab. Here the H basic 4 chain unit contain two separate but identical antigen binding site diversion. This is the antigen binding site, variable region here, and this constant region, the heavy chain, the hang region, SS bonds. Cl plus CH are attached by disulfide bonds. CH counterparts may be linked by disulfide bond. The region at the base of each arm of T or Y located between CH1 CH2 hang region. Loose secondary structures that make it flexible, more freely moving. Here, location of RSS in germline immunoglobulin DNA, lambda chain DNA, VLC, and this is kappa chain DNA, LVLC, the heavy chain DNA, VH, DH, GH, CH. This immunoglobulin chain differentiation and production. Here we find the leader sequence with blue, variable region in pink, diversity region green, joining LG region in red, constant region in green. VDG rearrangement, basal DNA VDG, combination variable recombination VG recombinant, mutation occur, papain act on an terminal side of enter H chain this disulfide Ig split into chain VH CH monovalent but for some engine fab fragment and binding CH terminal of both H chain disulfide identical for many different Ig crystallizable FC. It has secondary biological activities of engine recognized by FC receptor found on many types of cells. The enzymatic digestion product papain pepsin act CHO on CH terminal of inter H chain disulfide signal large fragment disulfide linked fab fragment bivalent engine binding activity. The immunoglobulin chain and the fragment formed by polymorphic digestion. Classification of Ig, light chain type, subtype. This individual divided into kappa and lambda on the basis of CL region sequence. No known function difference between kappa and lambda. Kappa is only 1, lambda is up to 6 different forms, functionally identical yet separate chromosome. Classification. Kappa to lambda equal to 2 to 1. A given Ig molecule always contain exclusive kappa or 1 of lambda. Heavy chain class subclass, weight about 50,000 to 7,000. 5 different classes, isotype, each with different physical and biological properties. This IgM, IgD, IgG, IgA, IgE. Normal individual express the 9 classes, subclasses. Each is enclosed by a separate genetic locus inherited independent. Here the IgG1, IgG2, IgG3, IgG4. Substructural difference in Igs lies in arrangement of interchain disulfide bridge within each 4 chain. LH bond is situated close to VHC-CH1 domain junction, close to H1-CH2 domain junction. IgG, IgD, IgE, IgA, IgM. Membrane bound secretory Igs. Here's the engine binding site, hypervariable region of heavy chain. Membrane bound Ig, individual 4 chain units. Each chain additional CH terminal sequence of 40 AA, 12 to 40 highly acidic region show little AA conservation, 26 AA hydrophobic transmembrane which anchor the H chain similar in all chains. This similarity is essential to bind to the same integral membrane protein Ig alpha, Ig beta in order to transduce signal into cells. One should differentiate between the Ig directly anchored to the cell membrane B cell lineage only and the indirect association of soluble antibody bound to Fc receptor on many cell types. Secreted IgS lack the terminal transmembrane segment as a result of RNA splicing. In this place, the secreted form of mu and alpha contain short terminal sequence tail piece which mediate polymerization of 4 chain units serve as contact site for G chain. Allotypic forms of H and L chain. H chain subclass L chain subtypes are encoded by separate genetic loci, single haploid genome. Alternative form allele differ by only one or few AA substitution allotypes. In human allotype in H chain, alpha and epsilon, gamma, alpha, epsilon, and in L chain, kappa only. Do the allotype have an effect on Ig function? Then why are we interested in this issue? Because this variation, Ig immunogenicity, immunization by pregnancy or blood transfusion, RF directed to normal Ig chain secretory component, G chain and secretory component, G chain small acidic protein not related to H or L chain, found only in polymeric Igs bounded by disulfide bond to cysteine tail segment in alpha and mu chains. About its function, it's not known exactly. Properties of human IgS, Igs, the Ig, IgA, IgM, IgD, IgE, the H chain class, L chain, the molecular formula, the electrophoresis mobility, the concentration, the placental transfer, mast cell or basophil segment, bacterial lysis, and the antiviral activity, IgA and bacterial lysis in IgM. Here, Hunter immunoglobulin unit, anti-IgA, patient, anti-IgG, anti-IgM, anti-kappa, anti-lambda, and the stain, and that's control and standard. Immunoglobulin G, 75% of total Ig being 70% Ig1, 20% Ig2, 60% Ig3, 40% Ig4, antibody of secondary response. Ig4 cannot fix C by classic pathway, but can do so via alternate pathway. Functions only Ig that cross placenta, so it's protect newborn during first few months of life. Fix complement, bind site reside in CH4 domain. Other cell express chain receptor which bind Fc region of IgG, especially Ig1, Ig3. Role of mucosal IgG. Some external secretion contain relatively high level of Ig, cervical mucus, vaginal fluid, serum, nasal secretion, can be of systemic respiratory tract or local region. Mucosal Ig can be induced by systemic or local intranasal immunization. Mucosal Ig induced by systemic immunization can be protective in influenza virus, conjugated polysaccharide vaccine. Immunoglobulin A, 10 to 50% of the total serum Ig. It's predominant in Bayer patches, tonsil, and other submucosal lymphoid tissue. Most abundant in secretion, saliva, teeth, intestinal mucus, bronchial secretion, milk. Ig1, IgA2 have similar properties. Forms in B cell surface monomers, one for chain unit, in blood secretion multimer to up to 5 units. The predominant secretory IgE dimers or trimers. Biosynthesis and distribution of human IgG. Immunoglobulin M, antibody of primary response, 10% of total serum IgE secreted as G chain containing pentamer, mostly commonly expressed on the B cell surface with IgT. One molecule of IgM fix complement very efficiently. Immunoglobulin D, monomer 180 kD, found in OB cell surface beside IgM, rarely secreted in significant amount. ID on cell bind antigen intercellular signaling consequence as IgM. After activation is finished, IgD expression cascade. Physiological function, antibody activity toward insulin, penicillin, milk, diphtheria toxoid, nuclear component, typhoid, thyroid antigens. Monomer of total serum level involved in allergic disorder, IgE. Immunoglobulin D, monomer 180 kD. Mast cell basophil carry Fc high affinity receptor with recognized CD3 CH3 domain of a absolute chain. These cells are always decorated with IgE absorbed from blood that serve as antigen receptor on them. Increase in allergic disorder, helminthic infestation. Immunoglobulin E, monomer of total serum level involved in allergic disorder. Mast cell basophil carry Fc high of high affinity receptor with recognized CD3 domain. These cells are always decorated with IgE absorbed from blood. Framework carbohydrate region, adenotype, V region subgroup. Immunoglobulin variable region. Hypervariable region, complementarity determining region. Here the antigen binding site, hypervariable region of heavy chain. Framework region, invariant stretch of 50 to 30 AA among the variable region. Hypervariable region, short region of extremely variable region. VH contain hypervariable region. Each sequence of these regions determine the antigen specificity, CDR1, CDR2, CDR3 from amino terminus, CDR3 longest and most variable. Schematic representation of the humanized antibodies, chimeric on the left and the fully reshaped on the right. In the case of the chimeric antibody, the rodent heavy V chain and the light chain variable region white are attached to human constant region black. After full reshaping, the 4 framework regions within each V region are humanized, leaving only the 6 complementary determinant region, 3 in the heavy chain and 3 in the light chain white. V region subgroup, sequence of many variable regions from any one type of chain form subgroup that are similar to one another than to the remaining V regions. Subgroup different in length and position, insertion and deletion. Idiotypes or self-type, unique V region AA sequence of homogeneous Ig produced by single B cell clone. Idiotype of B cell clone, antibody binding site in a given Ig determined by the hypervariable sequence of particular VH and VL domain. Anti-etiotype antibody recognized sequence in CDRS of target antibody which are unique to that antibody. Selected condition which have been associated with monoclonal antigen: macroglobulinemia of Waldenström, CLL lymphomas, benign monoclonal gammopathy, systemic capillary leak syndrome, amyloidosis, chronic liver disease, autoimmune disorder, Cushing's disease, malignancy of various type. Here polyclonal hyperimmunoglobulin, some associated disease state, immunodeficiency diseases, IG class, IgE, IgE, IgM, infection, congenital infection, IgM or all, tuberculosis, intestinal parasitism, liver diseases, biliary cirrhosis, IgM predominant, and lymphoid hepatitis, loboid hepatitis, all classes, pulmonary disorder, all classes, and autoimmune disorders, all classes in